we're just going to quickly review the study guide questions that we talked about last Thursday. And sorry, my PowerPoint's just taking a second to load. So we're going to go over the study guide questions that we went over last Thursday. So hopefully in your groups you were going over these, defining pH, describing the functional group of organic molecules. So instead of just watching me talk with my hands about these things and reviewing them, I actually created kind of a smaller subset of study guide questions and PowerPoints. And so I pulled directly from the PowerPoints that I had done a lecture to answer these study guide questions, and then I'll post the study guide question synopsis at the end of today's class. So again, like I said, define pH and the role of the buffer systems in homeostasis. So pH and the role of buffers, Dylan, the answer to that is yes. You're only present at one lecture a week. So you just need to go essentially 14 times across this semester. So hopefully that amount of flexibility helps you all out significantly. But if you are having, let's say, a little bit of an issue with a particular chapter, I mean, there's no harm in coming twice a week. So just please make sure that if you are having a hard time with the material, that you do come, because we're going to discuss it as a group. Thanks, Ethan. So in terms of defining pH, it's important to recognize that an acid is defined as a high number of hydrogen ions. So these types of molecules, you have the dissociation of those molecules, a positive and negative. And so that positive hydrogen, that a high number of hydrogens, is going to give you the acidic substance. And so that's going to raise the, it's going to lower the pH the greater number of those hydrogen ions you have present. For the bases, in this case, you have potassium hydroxide. It dissociates in water, where you have the release of a potassium ion and a hydroxide ion. And it's the high concentration of those hydroxide ions, that makes that substance basic. And then if we look at a basic salt solution, you have potassium chloride here, and those ions are dissociating, but you're not necessarily seeing a change in the pH. So this particular figure is actually really nice because what it does is it allows you to kind of see the differences in the amount of hydroxide and hydrogen ions and how that alters the pH. So it's important to probably recognize here at the when you have equal numbers of hydroxide and hydrogen ions, that you have that neutral pH. The greater number of hydrogen ions, it becomes increasingly acidic, and the increasing number of hydroxide ions that you have present, it becomes far more basic. And so this is again showing you kind of the interplay between these two ions and how the increase and decrease of each is going to affect your pH. So in terms of the body, like why is this important? There are going to be parts in your body that are going to be more acidic and more basic. For example, one place that you might find more acidic is where? I'm going to go ahead and open this up on chat really quick. Where would you find a really acidic environment in the human body? Nice, Hallie. Yep, exactly. So the stomach is going to be very acidic. Obviously, it's important for that acid. It assists in breaking down the proteins that you consume, and it's kind of, again, part of just a digestive process. But it is so important that the body maintains homeostasis. And so for that to occur, you have to have buffer systems available to buffer that pH. So in other areas of the body where you don't want these drastic shifts from basic to acidic, you have the presence of these buffers. And what buffers do is they kind of act as sinks, or kind of just soak up those hydrogen ions or those hydroxide ions. And so here we have one example of that would be sodium bicarbonate. And then this is a very similar example that we see here. So buffer systems are available. We've got other molecules available that are essentially soaking up those excess hydroxide or hydrogen ions. Okay, so before I move on from acid, bases, and buffer systems, are there any questions?
Would you mind saying the salt thing again really fast?
Okay, sure, yeah. So if we look at salts are just two ions of opposite charges that are attracted to one another. So the example that you see here are going to be potassium and chloride. Sodium and chloride are another example, because the sodium ion is positive and the chloride ion is negative. If we remember from last week, we were talking about ions. It's kind of, again, that complete shift of an electron from one to the other that gives them those absolute different charges. So if you put a salt into an aqueous solution, or water, it's going to break down into those charged ions. And so you're going to have that positively charged cation and that negatively charged anion. So I kind of went into a little bit more that times, but hopefully that kind of clarifies it. Are you good? Okay, yeah.
Yes, thank you very much.
Yeah, no problem. And at any point, feel free to, like I said, I talk very quickly, but I want to make sure we have enough time to get through everything we want to today. Okay, so question 2 is describe the functional groups of organic molecules, and this is basically taken straight from the lecture slides. So we're organic beings, right? We're made up of carbon, but we've got all these kind of molecular decorations that make these carbon chains of carbon slightly different. So we can have hydroxyls that if their OH group is attached to kind of a carbon structure, those can be considered alcohols. Molecules with OH groups dissolve more easily in water because, again, it creates more of a polar molecule, meaning a slightly negative and a slightly positive charge. A sulfohydryl a group on top of organic molecules are thiols. Again, this is very polar. And there are certain amino acids that actually have this group on there. And so we get to talk about proteins, and kind of what makes proteins different is that they have these groups that hang off of them. And for example, cysteine is one of them, and it's got a sulfohydryl group that hangs off of it. We talk about why this is important. There are proteins that have this three-dimensional protein structure, and cysteine, if you have two cysteines that are close together, they can form a disulfide bond, meaning two sulfur groups can actually form a bond between them, and it holds kind of the structure of that protein in its three-dimensional shape. Now, I don't expect you guys to know that to that level of detail, but it's just an example of kind of why we're learning these particular types of functional groups. A carbonyl group, ketones is a good example of that. Again, you're creating a polar situation on that carbon, and they're hydrophilic because you've got that polar characterization. So anytime you see something that is polar, always think hydrophilic. If it's polar, it's happy in water. If it is nonpolar, think of, okay, nonpolar, fats are nonpolar, they're very unhappy in water. So polar, and again, that's just that's something you'll probably see somewhere on an exam, because I think the polar nonpolar ideas are really important, and they were also, I think, part of your quiz over this chapter. Aldehydes have a carbonyl group in the end of the carbon skeleton. We see this in the case of the amino acid or the polypeptide chains. And then we can see here are also carboxyl groups. You have carboxylic acids, the end of the carbon skeleton, the COOH group. It's negatively charged, meaning that it's polar, again, more happy in those hydrophilic environments. Ester groups are dietary fats and oils, as you can see here, and also available as a molecule from the willow tree that is pain relieving. Again, we're kind of just going very quickly through some of these functional groups. And if anybody needs me to slow down or go over in another example, please make sure you're using your chat function, and I'm happy to do that. So phosphates, we see phosphates in the body primarily, I guess one place that you see it is in DNA, the phosphodiester backbone, phospho, meaning phosphate group. There's a phosphate group there that kind of links each one of those nucleotides together. It also has a role in ATP, or adenosine triphosphate, tri meaning three. There are three phosphate groups attached to an adenosine, and each one of those phosphate groups kind of holds energy in its bond. We'll talk about that a little bit later. So when each one of those bonds is broken, energy is released. So adenosine triphosphate is kind of the highest energy form of that molecule. And then if you lose a phosphate group, energy has been released, and you have adenosine diphosphate, meaning two. Aminos or amines, this NH2 group, another place that we see that is in the polypeptides or the protein structure. So proteins have a carboxyl group at one end, an amine structure at the other, and then there's a carbon in the center in which the R group hangs off of. We'll talk more about proteins here in a second, but that's, again, just probably the most clear example of where you're going to see an amino group for an organic compound. Okay, so before I move on from functional groups of organic compounds, this is not a chemistry class. I'm not going to ask you to draw these structures. I might throw out like one as an example, and the ones that I ask you about will be ones that we've seen in multiple places. So the phosphates, We're seeing that in ATP, ADP, so I might ask you about that one. The amino and the carboxyl, we're seeing that here, but then we're also seeing it when we talk about proteins. So I probably, if I was going to like highlight possible places or ones I might ask you about, I would say those would probably be the groups that I would ask you the most likely about because they're applying to different places within this chapter. Okay. Okay, if no one has any questions, then I'm going to go ahead and continue to move on. Okay. Okay, so identify the components and functions of carbohydrates. We can think of carbohydrates as sugars. So glucose is probably the carbohydrate that we'll be talking about the most. It's the one that gets entered into our metabolism and gets broken down. And so other sugars that don't look like glucose eventually end up getting broken down. So they do look like glucose, so they can be metabolized because the way our metabolism works is we have very specific enzymes that break down these sugars. And so it's important that they look like glucose before our metabolism actually functions. And so this is just an image of glucose. It's basically just showing you that glucose is this ring structure. And we'll talk about kind of how other structures might end up eventually looking like glucose. So the major carbohydrate groups, you have monosaccharides here. Monosaccharides are a single ring structure. So glucose, fructose, galactose, deoxyribose, and ribose are all considered monosaccharides because they do just have that single ring structure. So mono meaning one. And then you also have instances where we consume disaccharides, which are two. They're two ring structures. And you have two monosaccharides that end up getting joined together to form this two ring structure. So sucrose, lactose, and maltose are all examples of that. But you can see here that you have the combination of the two monosaccharides given here that make up this disaccharide. So like I said, for our metabolism to function correctly, these disaccharides have to be made into glucose to be able to enter the metabolic cycle. Or if you have another monosaccharide, let's say fructose or galactose, those monosaccharides have to be modified in some way by an enzyme to be made to look like glucose so it can enter our metabolism and be broken down to create ATP. Polysaccharides, so mono meaning one, di meaning two, poly meaning many. So exactly what you would think here, you have a huge string of monosaccharides that are kind of linked together. So glycogen and starch and cellulose are all examples of polysaccharides. Glycogen is essentially kind of how we store energy within our bodies. Starch is a form of carbohydrates in plants. And then cellulose, it's important to recognize again that our body has very specific enzymes to break down these different types of polysaccharides. And as being humans, we actually don't have the enzymes to break down the bonds that make cellulose. And so that's why we're not able to digest them, AKA corn, for example, has a ton of cellulose in it. And so we're not able to digest corn all the time. You can kind of put two and two together as to what the output of that is. Okay, so the building blocks of carbohydrates, like I said before, we've got monosaccharides. This is a great example of showing some of the structures that we just talked about. So they're pentoses and hexoses. Pentoses meaning that there are 5 carbons in those ring structures. Hexoses meaning that there are 6. I don't think we need to get down to that level of understanding for you because this is not a chemistry class. I'm not going to ask you to draw these structures. Ultimately, I would ask you if it's a monosaccharide, and if you knew that glucose, ribose, or deoxyribose was 1, I feel like that is a good level of understanding here. So those are all structures of monosaccharides. The disaccharides you can see here have those 2 ring structures that are linked here. This bond that occurs between monosaccharides, that's called a glycosidic bond. You know, you think of glucose, kind of sounds a little bit like that. A glycosidic bond is the bond that forms between these 2 monosaccharides to make this disaccharide, such as lactose or maltose. Okay, I'm gonna pause there again for questions to see if anybody has any. Make sure I'm not sucking up every second of this class. Any questions? Okay. So polysaccharides is the last example here. Here you can see that there's a glucose monomer. So each one of these tiny rings here is just a molecule of glucose that has been linked together with these glycosidic bonds. So these long branch structures, again, those polysaccharides that we talked about were things like starch, glycogen, cellulose, are all those long branching polysaccharide structures. Okay, so that kind of wraps up question 4. I'm going to keep rolling along unless somebody interrupts me via chat. Okay, so identify the components and functions of lipids in the body. So lipids are going to be fats. What's important to recognize here is we're kind of going slowly through all of the macromolecules. Lipids kind of look a little different than the carbohydrates, but if you think about fats, we also metabolize fat in our body, and it's ultimately kind of how those fats get broken down into those carbon structures that look similar to kind of carbohydrates that allow them to be metabolized. And so if we look at the types of lipids in the body, you've got fatty acids that are used to synthesize kind of other types of lipids, things like triglycerides and phospholipids, and we'll talk about what those each do. And those can be essentially broken down to generate ATP because they're entering the metabolism in a different part of the cycle. Triglycerides are important for protection, insulation, and storage. Phospholipid is a major component of all cell membranes. So phospholipids are part of that phospholipid bilayer, which we'll hopefully talk a little bit, I think maybe today. Yeah, today we'll talk about that in the study guide for chapter 3. Steroids are important for signaling, but they also play a role sometimes in cell membranes. We talked about how they influence cell membrane fluidity, and then bile salts are needed for digestion of dietary lipids, and this exists in the gallbladder. Other types of lipids, here you can see hormones. Again, these types of lipids that are used for signaling in the body. These hormones are going to help regulate metabolism. They're also going to be released in stress. So we can think of different types of stress hormones that can be released, things like cortisol. They also play an important role, again, signaling long distances for salt and water balance. Sex hormones, we can talk about estrogen and testosterone, stimulate reproductive functions and sexual characteristics. Those are also considered types of lipids. The lipids that we see up in this top part here are a little bit more linear. These hormones that we see down here are going to be more ring structures, again, kind of looking at the chemistry. Echinocyanidins have a diverse effect on modifying hormones, blood clotting, inflammation. They have a lot of different, again, signaling properties within the body. And then just other lipids worth mentioning are things like carotene and vitamin E, important for cellular function, things like the synthesis of vitamin A, which has a very important role. There's a large part of the world that actually has a vitamin A deficiency that can affect eye function. And so actually one way that some outreach, I guess, institutions have tried to mitigate that is by making a genetically modified rice that's got vitamin A in it. It's called golden rice. So in parts of the world where they're vitamin A deficient, this golden rice can actually help provide that particular vitamin since they're not ingesting it enough in their daily diet. Vitamin E is important for wound healing and preventing scar tissue, and maybe some of you have used that to prevent scar tissue. Okay. So let's keep rolling on here. This is the same slide we just saw. That as well. Oh, maybe not vitamin. We talked about vitamin K, important for blood clotting, and then lipoproteins. So again, kind of talking about the big general functions of lipids in the body. You can see it has a lot of different functions. Some of those functions are in signaling. It's important to recognize these because these are lipids. They're able to cross that plasma membrane very easily because they're lipids. They cross that fat, that hydrophobic part of the phospholipid bilayer very easily. So that's what makes them kind of a good signaling molecule. So this is just a good, I guess, slide just showing you the different structures of each of these. So the phospholipids here, we can see that has a hydrophobic tail and a hydrophilic head. So that tail's got those long chains of fats. All of them have hydrogens on them. It's very, very, very nonpolar. Because it's nonpolar, it's hydrophobic. It does not want to be around water. And then you have a polar head that has that charge structure in it. It has that phosphate group. It's hydrophilic. It's super happy hanging out with water. So we have the bilayer because you've got those two fatty lipid tails that are hanging out and facing one another. And then on each side of it, I don't know if this gesture really helps, but on each side of it, you've got these happy phosphate heads that are happy to hang out in water and be in the extracellular and intracellular environment. Triglycerides that we talked about are important for, storage of energy. You can see here the triglycerides have 3 legs, not surprisingly, 3 kind of legs, and they're attached with a glycerol group head here. But you can see that there are different lipid tails hanging out here. And those tails have different names depending on how many carbons are present there. So palmitic acid, stearic acid, and oleic acid are all different types of lipids, and again, the number of carbons that make them up is what kind of gives them different nomenclature. What's nice about this triglyceride is it shows you an example of what both a saturated and an unsaturated fatty acid looks like. If it has all of the hydrogens saturating every possible bond, that's a saturated fatty acid. And then if you have some hydrogen bonds that have not been saturated, so in the case here you've got a double bond and you're basically missing 2 hydrogens, that's an unsaturated fat. So it kind of kicks that leg out, kind of changes the structure of that lipid. And so when we talk about fats, when you think of the word fat, you might think of like butter and oil. Butter has more saturated fats, which allowed it to be tightly packed together, and unsaturated fats have that little kink that we see there in that leg and causes it to be far more fluid. So that's why you see the differences between butter and oil. Okay. So kind of talked about fatty acids there. And so in this other corner, I won't go any more into fatty acids. So any questions about lipids? Okay. Thanks for responding. I just want to make sure you guys are still out there, because sometimes it just feels like I'm talking into the void. So I appreciate the feedback. Okay, so moving on kind of through the macromolecules, we've talked about carbohydrates, we've talked about lipids, and now we'll talk about proteins. So proteins, like I said, if you've watched the lecture, you'll see that the cell is, a little protein-making factory, and so melting saturated fats, then make them unsaturated. You're starting to break down those bonds, and so that's what changes. Kind of, it's a phase shift that you're also creating with that. So you're breaking a lot of those bonds and kind of making it more liquid by raising that heat. So hopefully that makes sense, Emma. And if not, please follow up. So for proteins, there are a lot of different things that proteins do. Proteins essentially carry out a majority kind of the functions and the work of the cell. You can have structural proteins, regulatory proteins, and contractile proteins. As we're going through the body, we'll kind of see examples of each one of these. Structural proteins are going to create structure for the cell, or structure for tissues within the body. Regulatory proteins are going to allow the cell to respond to external cues in its environment. So a regulatory protein could be like a receptor that's supposed to sense a neurotransmitter. And so insulin is another example that's shown here. And again, it's just allowing the cell to respond to its environment. A contractile, an example is myosin and actin. Those are kind of structural proteins too, but they actually allow the muscles to contract and alter their shape. Other types of proteins are immunological. Antibodies are a great example of that. That's the way that your immune system is able to essentially recognize self from non-self, is by creating antibodies that detect those self and non-self molecules that enter into the body. So once you have a non-self, let's say a bacterium that enters the body, your immune system basically raises these antibodies against that, kind of as a, hey, this is not us. And those antibodies go out and try to find other versions of that. And if so, they find it, then you're going to have a mounted immune response. Transport, it's important that our bodies get oxygen to all of its tissues, and so hemoglobin is a great example of a transport molecule. It's a 4 subunit molecule that has an iron associated with it, and that allows oxygen to bind to that particular set of proteins and allows it to travel to all the distant locations within our human body. And catalytic proteins are often regarded as enzymes. Enzymes are proteins. Enzymes catalyze reactions and allow reactions within the body to occur with far less activation energy than they would otherwise use. So ATPase, sucralase are all, anytime you see a word in the body that has ASE, ACE in it, just a heads up here if you're taking other biology classes, ACE means that it's an enzyme. So just, you know, if you see an ACE, you're like, oh, it's an enzyme. That's what that protein does. Okay, so questions about components and functions of proteins. We'll talk about kind of what the actual structure of proteins are here in a second. Okay, I'm going to keep moving forward. So the building blocks of proteins are going to be amino acids. You'll notice here kind of there's a structure of like an individual subunit making a larger component. So amino acids are going to make up proteins. So we can kind of see the very basic structure of an amino acid here, where you have an amino group on one side. Hey, didn't we just talk about amino groups? Yes, we talked about it when we were talking about those side groups for carbons. Here we have a carboxylic acid. Again, kind of just know that structure. We just talked about it. And then you've got a central carbon here and then an R side chain. And that R is basically 20 possibilities, 20 different amino acids that can kind of get attached to that R group and change what the identity of that amino acid is. So there's 4 different examples here. These are just representative ones, but like I said, there's 20 total. So glycine is kind of the most simple amino acid because that R group is represented by this H. And you can also have cysteine that we talked about here just a second ago, where you have that sulfur group that's attached and that forms disulfide bonds within protein structure. And then you can get far more complicated amino acids that are represented by that R group. So here you see tyrosine and lysine as different representative amino acids. And so if you haven't looked at amino acids before, this might not make a ton of sense. So just kind of, again, recognize that there are 20 amino acids, essential or branched chain amino acids. You're talking, I guess, about both. Amino acids, I guess, would kind of all be essential depending on the part of the body. The cell's going to require all 20 amino acids to be able to function. And you do have some branched chain amino acids amongst those 20, but there are those 20 amino acids that just kind of make up the composition of all proteins. I am not going to ask you about memorizing the 20 amino acids. If you ever want to see a class freak out, you tell them they have to memorize the structure of 20 amino acids. Woo. So definitely not going to do that. But just recognize that their basic structure here is this amino group, this carbon, and a carboxylic acid. And then you kind of have an R group chain there that can be represented by 20 different other side chain structures. So how are these amino acids actually forming proteins? And you can see here that the two joining of these 2 amino acids down here are glycine and alanine. And basically it's what's called a dehydration synthesis, and you lose a molecule of water. So in this dehydration synthesis, a molecule of water is lost, represented by this green circle. Can you guys see my cursor? Okay, good. I was going to start annotating, which would probably make things, okay. So you can see this green bubble here is where you're going to be losing that molecule of water, and what ends up forming is a bond between that carbon and that nitrogen, and that bond is called a peptide bond. You'll recognize here that you still have an amine group here, you still have a carboxyl group here, but you've joined together the 2 amino acids to form a peptide bond, and that one R group is represented by that H, and that other R group is represented by that CH3. Again, you've just joined together glycine and alanine and made glycoalanine a dipeptide. Dipeptide, again, meaning 2, but you've lost water. Now, if you're wanting to, let's say, break peptide bonds, you would do the exact opposite. You would add water to the reaction, and that would cause hydrolysis, or breaking of something with water. This obviously doesn't occur with just the addition of water, or all of our proteins would just be dissociated. This bond is pretty strong and has to occur only under specific circumstances, sometimes mediated by an enzyme. Okay, so this is the building block of proteins, are amino acids. So the functional role of proteins, kind of what proteins do, I think we went over here a second ago. You know, we talked about how proteins have all of these different functions within the cell, provide protection, assist in muscle contraction. They do regulation and transport. Enzymes have a major function in the cell. Again, they're very specific. They catalyze all of the reactions that occur in the cell, which, you know, there's hundreds of thousands that are occurring in any cell at any given time. Enzymes, I'm just going to talk a little bit more specifically about them here. Enzymes are proteins that don't change or gain or lose anything during the reaction in which they catalyze. They just conformationally change, meaning that they change shape. So you'll have your two reactants or substrates come into kind of a little hole or whatever into that protein, and then that protein is just going to kind of change shape, help those two, you know, substrates interact or, you know, react in some way, and then it's basically going to change shape again and release those substrates, and they're going to be released into the environment. So it's kind of, you know, an interesting interaction that happens in which that protein is catalyzing those reactions. It's very efficient, it's very specific, and it can be controlled. So a lot of enzymes are actually, we just talked about phosphate groups. Enzymes are often are regulated by the addition or loss of phosphate groups. So those addition and losses of phosphate groups can act as on and off switches for enzymes. But it's not, that's not in the lecture notes, so I'm not going to expect you to know that. But just recognize that enzymes can be regulated by other cellular or other molecular mechanisms too. Okay, any questions about proteins before I move on to nucleic acids? Okay, so nucleic acids, again, describe the structure and functions of deoxyribonucleic acid, DNA, and ribonucleic acid. So these are going to be the molecules that are going to carry the genetic information for the cell. So DNA is actually going to be the thing that carries it, and then RNA is going to be kind of an intermediate molecule that kind of carries out some of DNA's functions kind of out in the cell. So RNA helps in protein formation. So if you've watched the lecture, you've maybe seen me draw the DNA becomes RNA becomes protein. That's called the central dogma of biology. That's kind of a cornerstone, or like the big picture of what happens in the cell is, you know, I talk about the cell being a protein making factory, and that's exactly how it happens. You've got your blueprint DNA that gets made into RNA, which is your intermediate, and that RNA is read out into protein. So that's just very basically what happens in the cell, and what kind of all the organelles are doing in the cell outside of carrying out the specific functions of maybe that cell. So we can see here, we'll talk more about what a nucleotide is made up, so we won't talk about that specifically now, but we talk about the differences between DNA and RNA. DNA is going to have cytosine and thymine, adenine and guanine, and then RNA is going to have uracil in replace of thymine. You'll recognize that the structures look very similar there. There's just subtle differences, but just that subtle difference allows people to detect the differences between DNA and RNA. If we look at the structure of DNA, it's a double helix, meaning that there's two parts of the staircase. It's almost like a twisted staircase. It's an alpha helix is the structure of DNA. And the thing that holds like the stairs of that alpha helix together are hydrogen bonds between those nitrogenous bases. And so those nitrogenous bases have selectively paired to one another. So only thymine will pair with adenine, only cytosine will pair with guanine. And so that pairing is very specific. You'll see that there are different numbers of hydrogen bonds between these pairings. You'll see 2 hydrogen bonds between thymine and adenine, and 3 hydrogen bonds between cytosine and guanine. So again, just very quickly, what's interesting, again, a difference between DNA and RNA is DNA exists most of the time primarily in a double-stranded structure, and RNA can exist in a double-stranded or single-stranded structure. The single-stranded structure of RNA allows it to carry out a lot more functions. RNA actually has the ability to act as an enzyme as well. So it's a very multifunctional molecule, RNA is. And again, because it's single-stranded, tends to also be maybe a little less stable and can be degraded by the cell a little bit more easily. So distinguish between DNA and RNA, well, we just talked about the differences in base pairing. Again, you've got the substitution of uracil for thymine. The sugar in that sugar phosphate backbone is going to be different for those backbones too, so deoxyribose versus ribose. Essentially, there's an addition of a hydroxyl group on one of those structures, but I don't need you to get down to that level of chemistry. Number of strands, DNA is 2, RNA is 1, nitrogenous basis. Again, we talked about A with T, G with C is for DNA, A with U, and G with C is RNA. How is it copy? DNA copies itself, and then RNA requires DNA as a blueprint. Again, DNA is always kind of the basic structure, the basic blueprint that all other, RNA is made from. Yeah, there are, because this is biology, I know that there are exceptions to some of these rules, but I'm not going to tell you about that now because you don't need to be confused by that. But again, because it's biology, there's almost an exception of every rule here. I mean, there are instances where RNA can be used as a template to make DNA, but we don't need to talk about that today. Okay, so functions for DNA is information to make proteins. RNA actually carries that out. So again, it's an intermediary between DNA and proteins. That RNA, or that we'll say more specifically the messenger RNA, is read by ribosomes and makes proteins. If you haven't watched the chapter 3 lecture, we'll talk about that more there. And then DNA exists in the nucleus in the mitochondria. If anyone ever wants to talk about why mitochondria has their own set of DNA, it's actually kind of cool. Used to be kind of its own cell, or that's part of the hypothesis. And then some of the functions, types, and then types of RNA, messenger, transfer, ribosomal RNA, all have different functions in creating proteins. Okay, any questions about DNA and RNA? Oh man, I'm gonna have to blast through the rest of this quickly. Okay. So a nucleotide, if we look at what a nucleotide is composed of, you've got a phosphate group, a pentose sugar that is composed of either deoxyribose or ribose, and then the nitrogenous base, which you can see here can either be cytosine, thymine, uracil. I guess what I haven't talked about yet is the fact that there are different nomenclatures for the single ring or double ring structure of the nitrogenous bases. The single rings are the pyrimidines, the double rings are the purines, and the way that I remember this, and please come up with whatever way makes sense to you. I think of the pyrimidines. So a diamond looks kind of like a pyramid, and what do you do to a diamond? You cut it, or C-U-T. Again, that's the way the connection happened in my brain at some point in time. So C-U-T, you cut a diamond looks kind of like a pyramid, and by process of elimination, you know the purines are adenine and guanine. Whatever makes this information work for you. So just be kind of creative with some of your analogies or metaphors or anagrams. Okay, I think this is our last slide here. Describe the functional role of adenosine triphosphate. The role of adenosine triphosphate, as we talked about earlier, is for energy storage. Each one of these phosphate groups here, one of these bonds has a ton of energy in it. So when those bonds are broken, energy is released. And so when you add those phosphate groups in, energy is needed for that process. So we've seen some of these structures before, right? We've seen adenine before, ribose is part of the structure. So from the cell's perspective, you can see how some of these molecules and structures maybe are interchangeable for different functions, right? But here you can see adenine and ribose with the addition of 3 phosphate groups making adenosine triphosphate. And if one of those phosphate groups, you have adenosine diphosphate. And then if you lose 2, what do you have? I'm going to open it up because I've been talking for a while. What do you have if you lose 2 phosphate groups off of ATP? Adenosine, what phosphate? So you're going to have lost 2 phosphate groups. So
diphosphate.
So if you lose 1, so you have 3 for your tri. If you have 2 phosphate groups, then you have di. If you have lost 2 phosphate groups and you only have 1 phosphate group hanging out, you have adenosine monophosphate. So tri, di, mono. Does that make sense?
Yeah, yep.
Awesome. Yeah. And honestly, I mad props to you folks who are speaking up, who are chatting in. Like, I appreciate the interaction. Okay, so I'm going to post that kind of shortened version